Hello friends, how are you? Hope all are fine. Welcome to my channel Sam Biology Class. Today we will discuss about conventional polymerase chain reaction. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Polymerase chain reaction, also known as PCR, is a powerful technique widely used to make millions to billions of copies of DNA sequence. This technique was developed in 1983 by American biochemist Carey Mullis while working at Cetus Corporation in America. For this work, Mullis, along with Michael Smith, who had developed other methods of DNA manipulation, were jointly awarded Nobel Prize in Chemistry in 1993. Polymerase chain reaction that amplifies specific DNA sequences in vitro by using a series of temperature cycles and then analyze the amplified products known as amplicons using method like gel electrophoresis is known as standard or conventional polymerase chain reaction. By this technique, copies of very small amount of DNA sequences are exponentially amplified in a series of cycles of temperature change. Majority of PCR methods are based on thermal cycling. Thermal cycling. The process of thermal cycling exposes reagents to repeated cycles of heating and cooling to carry out different temperature dependent reactions, more specifically DNA melting, and enzyme driven DNA synthesis. Most PCR applications employ a thermostable DNA polymerase such as Taq polymerase. This enzyme was originally isolated from a thermophilic bacterium known as Thermus aquaticus. Most PCR methods amplify DNA fragments that are between 0.1 and 10 kilobase pairs long. A basic PCR setup requires several components and reagents. First one is a DNA template. The template contains the target sequence which has to be amplified. Number 2 is the enzyme DNA polymerase. This enzyme polymerizes new DNA strand. Thermostable DNA polymerase such as Taq polymerase is commonly used in most PCR applications. Number 3, 2 DNA primers. Primers are short oligonucleotides about 20 nucleotides long. Out of two DNA primers, the one primer is complementary to the 3' end of the same strand, and the other primer is complementary to the 3' end of the antisense strand. Without primer, DNA polymerase cannot initiate DNA synthesis. The primer provides the free 3' hydroxyl group to which nucleotides are successively added by the enzyme DNA polymerase. Fourth one is deoxyribonucleoside triphosphates that include DATP, deoxyadenosine triphosphate, deoxycytidine triphosphate, DCTP, and deoxythymidine triphosphate, DTP, and deoxyguanidine triphosphate. These are the building blocks from which DNA is synthesized by DNA polymerase. Fifth one is a buffer solution. Buffer solution. The buffer provides a suitable chemical environment for optimum activity and stability of DNA polymerase. Last one is divalent metal cations. Divalent metal cations. Ions such as magnesium ion or manganese ion are commonly used in polymerase chain reaction. PCR is generally carried out in small reaction tubes in a thermal cycler. In a thermal cycler, the tubes contain all components and reagents including DNA template, tag polymerase, DNA primers, deoxynucleoside triphosphates, buffer solution, and bivalent ions required for DNA polymerization. The thermal cycler heats and cools the reaction tubes to achieve temperatures required at different steps of the reaction. Typically, a PCR consists of a series of 20 to 40 repeated temperature changes. 20 to 40 repeated temperature changes called thermal cycles. Each cycle consists of 2 or 3 different temperature steps. The basic steps of PCR are as follows. Number 1, denaturation. Number 2, annealing. Number 2, annealing. And number 3, elongation. The temperature used and the length of time that applied at different steps of PCR are dependent on a variety of parameters including the enzyme used for DNA synthesis, the concentration of bivalent ions and deoxynucleoside triphosphates, and the melting temperature of primers. For example, This is the DNA template. This is the DNA template which consists of two DNA strands that run in opposite direction. These two strands are hydrogen bonded. First step of polymerase chain reaction is denaturation, also known as melting. In this step, the reaction chamber, which contains the reaction tubes, is heated to a temperature of 94 to 98 degrees centigrade for 20 to 30 seconds. At this higher temperature, the double-stranded DNA template is denatured or melted due to the breaking down of hydrogen bonds between complementary bases, thereby yielding two separate single-stranded DNA molecules. Second step is primer annealing. Primers are short oligonucleotides containing about 20 nucleotides. In this step, the reaction temperature is lowered to 50 to 65 degrees centigrade for 20 to 40 seconds. At this temperature, primers which are in excess in the reaction tube are annealed or base paired to the 3' end of the single-stranded DNA molecule. The function of the primer is to provide free 3' hydroxyl group to which successive nucleotides are added by the enzyme Taq polymerase. Third step is elongation. In this step, the primers are extended in 5' to 3' direction. For elongation, the reaction temperature is adjusted to 75 to 80 degrees centigrade. At this temperature, Taq polymerase copy the DNA template strand by adding successive nucleotides to the free 3' hydroxyl group, thereby synthesizing the complementary DNA strand in 5' to 3' direction. The process of denaturation, annealing and elongation constitutes one cycle. In one cycle, from a template, two DNA copies are obtained. Multiple cycles are required to amplify DNA sequences to get millions of copies. The number of DNA copies that are obtained after a given number of cycles can be calculated by the formula 2 to the power n, where n is the number of cycles. N is the number of cycles. If the process will continue for 30 cycles from one DNA template, such a huge number of DNA copies are obtained. So friends, in this way, DNA sequences are amplified exponentially to yield millions of copies by polymerase chain reaction. In conventional PCR, the products called as amplicons are analyzed by methods such as agarose gel electrophoresis. Gel electrophoresis separates DNA fragments based on their size. The size of amplicons is determined by comparison with DNA ladders that are also run on the same gel alongside the PCR products. DNA ladders are molecular weight markers containing DNA fragments of known size. Polymerase chain reaction has several applications including diagnostics and disease detection, forensic and DNA profiling, research and development, paternity testing, ancient DNA analysis and drug development. So friends, this is all about standard or conventional PCR. For more updates, subscribe my YouTube channel Sam Biology Class.